Welcome to all the speakers and the participants to the session on physical approaches to help bring nanomedicine to the clinic. And our first speaker this morning is Dr. Silke Krol from the nice place in Italy. Hopefully weather is good. So her presentation will be realistic in vitro models as way to real individual medicines. Before you start, I wanted to ask you, probably we can do two things. You can either send the questions to the individual speakers to me, or we have about 30 minutes. So you can also note your questions and we can discuss them together after all the lectures. So we can go both ways. So Mrs. Krol, I think now you can start.
Thank you very much. So I will start sharing my screen. Okay. You see my screen? Good, very good. So one of the major problems in developing nanodrugs I discovered was that we don't have realistic models. We are going in 2D cell culture and then very quickly, let's say, into animal experiments because we don't get the answers we are looking for. And this was the starting point, thinking about 3D models, but 3D models is also, let's say, quite an interesting system. So the idea is what we need is perhaps, in order to avoid also animal experiments, realistic in vitro models, which then can serve perhaps even as a platform for every single patient to choose the right drugs for his tumors, which is not what we are doing with personalized medicine at the moment, as you already know. In this context, I have to make a disclaimer because I'm co-founder of Encytos, which is a startup which will commercialize the spheroid substrates, or the platform I'm introducing now for cell culture and especially for realistic.
Ms. Krol, sorry, we can see your PowerPoint. We cannot see the full screen presentation.
Okay. Yeah. We can try again. Can you see the second screen? Can you see the screen?
No, we are still in the open PowerPoint.
Okay. Can you see the screen now?
It's still small. Okay, now it's getting better.
It looks like the presentation mode is not properly working. From time to time, I have this problem. If it's okay from the size, I would continue in this mode. Okay, so as you know, at the moment we are trying to personalize medicine, but still it's very often one treatment fits all, not all, but at least specialized or individual groups which are getting a therapy. Very often you have therapy failure in first line treatment, then you lose time and you have to go to second line treatment. And for that reason, it could be good if beforehand you already know if a treatment will work in the patient. At the moment, what we are doing is, this is something we are forced more or less to do in rare tumors where no approved treatment is available. In this case, we have to test different treatments to see what could help the patient. And in this case, we are suffering from the time lag between, let's say, diagnosis of tumor and the time before we can suggest the treatment to the medical doctor or to the oncologist for treatment. So the other problem also in the development of drugs and later in clinical trials is that there are a lot of factors are responsible for drug failure or which are influencing the efficacy of drugs, which cannot be covered in clinical trials. So there is, on one hand, the aspect of the genetics, which we are trying to cover by personalized medicine, for example, identifying breast cancer patients with HER2 biomarkers on the surface in order to have a more specific treatment option. Then age is influencing the response to drug, specific nutrition, health status, comorbidities, environmental exposures, and in this case, environmental could be also life circumstances. For example, smoking is identified as a factor which is strongly influencing the outcome of chemotherapy. Then there are some epigenetic factors and other therapies. Yes. So, but the future medicine will be personalized medicine on which everybody of us can agree. I already mentioned this. I will skip it, otherwise I'm running out of time. Models for starting development of drugs are usually the standard 2D cell culture, and we all know that this is a model which is not really realistic, because if you are forcing let's say, cancer cells to grow in 2D, the cells are modified, and the response you get most probably is not predictive of the outcome in the body. Then we decided that 3D organoid models are the better choice, and this is definitively true, because we already know that the drug response of a spheroid of a 3D cell cluster is very different from that one on a 2D cell culture. So we are usually developing 3D models, but there are some difficulties also there. And then we had Matrigel in order to mimic also the microenvironment in terms of having environmental tissue. However, the building of a 3D model is usually quite time consuming. And then our final choice is Xenografts, the so-called PDX, patient-derived xenografts in immunodeficient mice. But we still know that there is an influence also of the immune system, and also this is a very time-consuming process. However, we need this on one hand for clinical applications. We need 3D also for regenerative medicine, for example, transplantation of neurospheres in treatment of neurodegenerative disease. And we need them in vitro studies, let's say in the drug discovery process, or even to understand tumor growth or tumor development in the basics. What is needed is, okay, on one hand, we agree the best model for tumor treatment is when we are taking the tumor tissue. Then we need a short growth time in order to be able to have a very short time lapse between diagnosis and the initial treatment. We need models that are mimicking reality best without using animals, because we also agree that mice are not men, and so also partially the model in vivo is in mice is not predictive necessarily for humans, and this is why a lot of drugs are perhaps failing in clinical trials. And it could be good if we just have already a mixture of approved drugs, because we all know that approving new drugs for treatment usually needs a long time, and for that reason, if you can find the optimal choice and combination of drugs for this patient's tumor, it would be optimal. So what we have is inorganic topographic amorphous silica structure, which we have either in square conformation or in hexagonal conformation. We can create these structures with increasing complexity. So we, at a certain point, we have a fractal-like structure. And what we observed is that when we put primary tumor cells on top of this, we find on flat surfaces what you are used to see, let's say this flat pancake-like cells with a lot of stress fibers. When we put them on this, let's say, pyramidal structures, what we observed is that on one hand, we have a background of 2D, and we are creating spheroids. They are forming spontaneously, and they are forming usually within 24 hours. Then, when you are going to the more complex structures, you see that we don't get any more spheroidal or spheroids. We only get 2D structures. And let's say the more complex the system gets, the less cells, at least from primary tumors, we get. We analyzed then this microtumor, which you cannot see because it's covered by this. Okay. So we analyzed this microtumor, and as you could see here, we have inside a diffuse Darby signal, meaning that we have necrosis. We have a viable outermost cell layer. We see when we are going for alpha-SMA, this is a hepatocarcinoma, and what we see is that this underlying 2D layer is red for alpha-SMA, meaning that these are cancer-associated fibroblasts. We see a layer of cancer-associated fibroblasts, let's say, surrounding the microtumor, but the microtumor itself is positive in this case for alpha-fetoprotein, so it's made from tumor cells, as expected. We did also a 3D reconstruction of this microtumor, and what we observed is actually that this microtumor, this spheroid of tumor cells, is also enwrapped by a layer of cancer-associated fibroblasts. So, as I said, this formation on the surface of spheroids is quite quickly. It starts at day 1, and we get quite huge, quite well-defined microtumors at 6 days. However, they are already ready in, let's say, on day 1. And we did an experiment for a collaborator on a glioblastoma, which usually needs him to create spheroids 3 weeks in an expensive medium with a lot of growth factors. In our case, we needed 18 hours on the surface, and we got similar spheroids as him. Then we tested also with cancer cell lines. By the way, we did this with different primary tumors, so it was a proof, or we could prove that tumor formation was observed for glioblastoma, even for types of glioblastoma which usually don't grow in cell culture and which are not forming spheroids. We could do this with prostate cancer, and we got spheroids, and we tested also with pituitary glands, and we got huge microtumors. Then we tested, as I said, with cell lines. In this case, we did with a hepatocarcinoma cell line, and in this case, we compared the cell line on our surface, and we got spheroids in 4 days, while when you try it in Matrigel, we had to wait for 13 days in order to get the comparable size. CACO2 cells are also forming spheroids. Okay, this is unfortunately because of the not presentation mode. So we differentiated 8 adipose-derived stem cells into neurospheres. They were Nestin positive within 24 hours without any addition of growth factors. We built up on the surface on the very same surface an in vitro intestinal model differentiating HT29 cells, which were differentiated not with MTX, but within 6 days. We could differentiate them on the surface. They were starting more or less immediately producing mucus on the surface, and then we analyze them. Usually when you do methotrexate differentiation of HT29 cells, you need to incubate them in MTX containing medium for 30 days in order to push the differentiation. As I said, in our case, within 6 days we could differentiate. We tested them for mucus 2, to identify goblet cells for lysozyme, and as you could see, the differentiation is mainly in Paneth cells expressing a signal for lysozyme. We had some cells which were positive for Velin, and as I said, we had mucus, but this means that 3 out of 4 intestinal cells we got within 6 days, plus the mucus, instead of waiting for 30 days and having a much more complex model. So on our surface, on the same surface, you could do spheroid culture, you can have 2D and 3D culture, and our idea is that these cancer-associated fibroblasts are actually doing what they would do with healthy tissues, so they are invading. When you go to the more complex one, we found, as you already saw, that tumor cells are excluded, which means we have a one-step possibility to isolate cancer-associated fibroblasts in more detailed analysis, and we can differentiate cells on the surface. We showed this with adipose-derived stem cells, which actually in 1 week differentiate in neurons without additional differentiation factors. We are the following people. This is my team here in Bellis, in Castellano, Matteo and Francesco. The first experiments were done with Simona, and these are my both partners in Encytos. And if you want more information, just to go to our website. Thank you for your attention. Now I cannot hear you.
Okay. Sorry, I just was saying that I gave you 5 more minutes because we had some technical issues, so we should be definitely fair. Are there any questions to Dr. Krol just at the moment, or we handle them afterwards in the discussion session? Looks like no. So then at the present time, so then we just go to the next presentation from Professor Viola Vogel. From the ETH in Zurich, and her title this morning is appreciating and exploiting the mechanical design of proteins.
So good morning to everybody.
Good morning.
For decades now, we have exploited the biochemical design of proteins, but not the mechanical design of proteins. So why is looking into this question important? One of the biggest challenges currently in tissue engineering and regenerative medicine is to learn how the body orchestrates functional tissue repair versus disease progression. And while we learned so much biology from 2D cell culture, the real niches in organ tissues are 3-dimensional and full of cells of all kinds of different cells, including fibroblasts, myofibroblasts, cancer stem cells, cancer cells, immune cells, including macrophages, and then certainly highly important, the endothelial cells. So where are the huge knowledge gaps that hamper faster progress? So mostly we've exploited either for the rational drug design or for high throughput drug screening efforts, the equilibrium structures of proteins. We learned so much about the structure of proteins from textbooks, and you see many beautiful examples on the left. Yet these structures and the high resolution structures are all equilibrium structures, either derived from X-ray or from NMR. So what was understood very well is how proteins fold along a very complex energy landscape. And for a long time, we thought mostly folded proteins have an important role to play in biology. And particularly in the lab, intermediates were often not even accessible because they were so short-lived and therefore not accessible for drug screening applications. However, imagine that you take a protein and pull on it, then you might stabilize some of these intermediate states that otherwise would not be seen. So proteins and motor proteins are well described today. We understand how they convert chemical energy into mechanical energy, but by doing so, they can apply forces to their intermediate filaments and therefore also to all the proteins and protein networks that are connected to them. So the first insights that proteins have fantastic mechanical properties that are different from polymers emerged when Hermann Gaub's group in 1997 started pulling on single proteins. And the unfolding characteristics were so different from what was expected from polymers, but proteins have secondary structure. So immediately after seeing these experiments, we started a collaboration with Klaus Schulten to ask what is happening structurally? What needs to happen in order for protein's equilibrium structure to break apart? And here you see the cover of the Biophysical Journal, and these were the first steered molecular dynamics simulations considering explicit water molecules and how they interact with the proteins as they are torn apart. One of the key findings was that thermodynamical stability is not equal mechanical stability, because as you pull a protein apart, force-bearing protein clusters of hydrogen bonds need to be broken. And there it makes a huge difference how you break these clusters of backbone hydrogen, whether in a zipper mode or in a chair geometry. So how can we utilize such information? First of all, it was thought that proteins only serve as shock absorbers. But then we developed the first non-equilibrium structures, illustrating that you might actually change not only the structure, but also the function of proteins. Here illustrated by pulling the type 310 domain of fibronectin apart that exposes the RGD loop to cells. So to test these first structural models, we developed fiber stretch assays. We put droplets of high concentration fibronectin, pulled fibers out of those and deposited these fibers on stretchable substrates. And these stretch assays gave us huge information about how the function of these fibers is changing as we stretch them. Why is this even relevant? This is relevant because fibronectin not only exposes the RGD loop, but many, many binding sites to growth factors, to other extracellular matrix proteins. It has a number of cleavage sites and is recognized by a variety of integrants. So over the course of the last years, we tested out many of these functional sites and could indeed show experimentally also that they are either activated or destroyed by mechanical forces, by stretching fibronectin fibers. So very recently, we found now, and wound site repair always starts with blood clots, and it was thought that the key role of platelets is to stop the bleeding. But we find now that platelets are also the first players to assemble new extracellular matrix. In order to visualize that, we used super resolution microscopy, here storm imaging, and indeed you see platelets over here and how they start pulling the first fibronectin fibers. So far, it was thought that mostly invading fibroblasts would form these fibers. But with platelets forming the first fibronectin fibers, they essentially create anchoring sites for other cells to invade blood clots. And most excitingly, they do so in contrast to fibroblasts, not by exploiting alpha-5 beta-1 integrins, but alpha-2b beta-3, which also recognizes the synergy site. So interestingly, so much has been learned in the last 2 decades now at the single molecule and the cellular level, how cells exploit these non-equilibrium structures of proteins in order to recognize the environment and material properties. However, at the tissue level, we have so many observations that mechanical forces in physical environments really matter, but we do not have a probe to either measure at the nanoscale forces applied to the environment or the molecular strains that might characterize tissues. So in order to address this gap, because it's a major limitation for us to ask how mechanobiology changes either during development or under disease conditions. So here we take now advantage of a beautiful observation we made about 10 years ago, initially again with steered molecular dynamics, where we found that the adhesins that Assays utilizes to bind to fibronectin fibers in wound sites, that they recognize the relaxed structure, but not a stretched protein. They do so by utilizing multivalent binding motif. And this multivalent binding motif, and you see it beautifully in the simulation, is destroyed if you stretch fibronectin, here shown in gray. So with this, we asked, can we utilize these bacterial adhesins now as probe to study the tensional state of fibronectin fibers in healthy and diseased tissues? And to our huge surprise, and since nothing is known about the tensional state of fibers in tissue, we photo-labeled the peptide and then co-stained with a polyclonal antibody against fibronectin. And indeed, we see with most of the healthy organs that we studied in cryosections, we see they are full of fibronectin fibers. However, in healthy organs, very few peptide is binding. And that implicates that in healthy organs, fibronectin is kept under high tension. In great contrast to that, here you see that tumor tissues stain heavily for the peptide. And that means, and this is against our thinking that was put forward in the recent years, fibronectin fibers in tumor tissue are progressively more relaxed, either because of the presence of proteolytic enzymes, but also we see high correlations with myofibroblast, wherever we see that the peptides are binding. And myofibroblasts produce a lot of matrix, including collagen. So in locations where fibronectin fibers are relaxed, we see thick bundles of collagen fibers using second harmonic generation. And even more exciting, we can label this peptide, inject it into the vein of a mouse, and it goes through the blood circulation and it accumulates in tumor tissue, as shown here by radioactive labeling and imaging. So this is a beautiful collaboration that we have established with Martin Beer at the PSI in Switzerland. So we hope to develop now this probe as a new nanoscale probe by exploiting multivalent binding motifs to probe the tensional state of proteins at the tissue level and to utilize these probes for diagnostic applications. So in order now to test and go beyond correlations at the tissue level, we are building de novo grown 3D model tissues. Here you see we are seeding fibroblasts on engineered clefts, and they start filling the edges with tissue. And here you see the growth over a 9-day period. And we make very interesting observation as the growth front is filled with myofibroblasts that convert back to fibroblasts as these tissues mature. So building up these microtissues is crucial to learn and develop causal relationships. Why is this all important? Currently, there are very few drugs that target non-equilibrium states of proteins, yet non-equilibrium states are heavily exploited by nature to regulate function. And in cancer therapy, we totally focus on killing the cancer cells, but we leave the diseased extracellular matrix behind. So with this, I would like to very much thank you for your attention, my research group, for all the beautiful work that they've done over the last years and decades. Thank you.
Thank you very much, for Professor Vogel. I really liked your animations. Very great, great work. Any questions from the other colleagues? Maybe too early for questions. Okay, but we still have time later on. So at that moment, I think we go from Zurich to St. Gallen to Dr. Peter Wick. His presentation is biomedical nanoparticles biotransformation analysis of an early dynamic event in situ. So Peter Wick is working in St. Gallen, as I said before, at the Empire Institute.
Yeah, thank you very much for the kind introduction, Marieluise. It's a great pleasure for me to be here, and I want to thank the organizers, in particular Beat, give me the opportunity to share our newest results. And I'm happy that I have Viola as a previous introducer of proteins, because I will also focus on early events, how proteins and particles will interact. So I have a similar technical issue. Now it goes. So I don't think so that I have to introduce in this audience nanoparticle and their power in therapy, diagnosis, as well as separation. We have learned a lot over the decades how to design particles with different architecture, with different functionalization, and we have even learned to rationally design these particles for a particular purpose, ending up with very nice suspensions, like here a gold particle suspension, which are perfectly stable, which we have under full control, and this buffer system allows to keep this particle suspension even over months in our shelves without really having significant changes. This changed dramatically if we transfer these particles into a biological environment. And long before this particle will reach the cells, a lot of things will happening as soon as they get in contact with a biological environment like full blood or whatever complex environment. So starting with a well-defined particle suspension could end up in relatively short time in a broad mixture of different modified particle originated from this original suspension. I just put here a sketch from a publication we published recently, Rethinking Nanotoxicology, that this particle could start to dissolute, that could get covered with proteins, that could aggregate, and sometimes it's not even clear what happened first or if this happening at the same time, but we know that this modification will have definitely an impact on the future fate of this particle in, for example, in the interaction of single cells or even a tissue. So here, the classical biochemical analysis comes to his limitation, and we have difficulty to follow these really early events in situ, label-free, and kind of an online monitoring. And exactly this challenge we looked at in order to bring light into this still kind of a black box in regard to understand these fundamental mechanisms. So for that, we created we put together a little library of gold particle from 5 nanometer in size, which are either citrate stabilized or pegylated, and a counterpart of 40 nanometer gold particle as a particle system, which we can well control. Then on the other side, we simplify the biological environment in that way that we only reduced the biological environment to one single protein. This is human serum albumin, but we use different concentration to mimic the different ratio of particle versus protein. And you see here that we have used the concentration of the protein with a ratio of 1 to 100 compared to the particle 1 to 1000 and over that. And we wanted to monitor what happens if we transfer this particle suspension into this NaCl solution containing this protein, what happens in regard of nanoparticle agglomeration, precipitation, binding, and or agglomeration, and so on. And we particularly focused on the very early time point from time point 0 to a few minutes. We reached that by using a small angle X-ray analysis, which is a wonderful technique allowing us to analyze particles, in particular very small particles, in a label-free, non-destructive way. And we can learn out from the scattered signal about the size, the shape, the surface, and also to a certain extent the interaction. And in order to have a good approach, we developed this microfluidic device, which has a first part, which is a kind of a classical mixing device where two fluids came together. In the middle, we have the particle solution, and from top and from bottom, the mixing buffer system is added. This will be mixed here, and then in this capillary, which we have a flow of a few microliter per minute, we can actually dissociate these early events by just analyzing with the X-ray beam early in this mixing device or late in this capillary, and we can actually dissociate this early fraction and later fraction very nicely. And we did kind of do the analysis on different position on this capillary, and by that we could learn what happens over time. So a particular result looks, first I want to show you the real machine in the lab. So we have here on the left side the X-ray source, we have here the microfluidic pumps. Here we have kind of our chamber where the microfluidic device, this mixing device, and the capillary are in, where the beam is actually then positioned. And here we have at the end the detector. And with that, we can actually do most of the analysis I'm showing in the future. So this is a particular result using 5 nanometer gold particle, which are pegylated in NCl solution, like a physiological ionic strength environment. And we see if we analyze the particle suspension early after the mixing device, we get this nice curve here, showing that our particles still remain in suspension. Over time, and these are these time points here. So this distance represents approximately 100 seconds. We see that here we have a shift in our curves, indicating that the particles start to self-organize themselves. So this indicates that we are sensitive enough to see small changes over this time. To make a long story short, I just put here as a sketch the results what we get. So we have here the analysis I showed just before that are without the protein, but with a low amount of salt. These pegylated 5 nanometer gold particles start to cluster to make this super lattice structure with a particle-to-particle distance of 20.7 nanometer calculated with a certain error. If we add then the human serum albumin, and we have here the low level, so meaning 100 proteins per particle, we have a slight shift in the superlattice structure where the distance from particle to particle becomes slightly bigger. And if we increase the protein more and more, we have also a tendency that this particle-to-particle distance become bigger. And we have also evidence that in the superlattice structure, this albumin, due to the fact that this has more or less the same size as our particle, might replace or be integrated in the superlattice structure, which is quite exciting to know and might be also open a new thinking or new view on nanoparticle stability in more complex system. As a counterpart, we did the same experiments with citrate stabilized gold particle. And as soon as we add the salt, immediately we have precipitations, and even the measurement does not allow exactly anymore what happened. So we have only very early event, and afterwards everything precipitate in the capillary, and the measurement becomes very difficult. Whereas already small amount of protein helps to stabilize the citrate stabilized gold particle suspension ongoing, and we might see in later stage that we have kind of hybrid formation between particle and the protein in a not well-defined structure. So sometimes we see particles be very close to each other, sometimes not. So here we have probably an arbitrary distribution of hybrid systems of protein and particle. As a further control, I would like to show you also the results of the 40 nanometer gold particle. And here we have kind of a similar effect. So the pegylated gold particle remains stable, even adding or having proteins in the environment. So that works very well. Whereas if you have citrate stabilized gold particle in salt solution, also here we observe an agglomeration and even a precipitation of the agglomerated gold particle. Low amount of albumin does not prevent this agglomeration. We need really high amount of proteins, which is more or less the situation as we have in the full blood. So this data we, of course, try to verify with cryo-TM analysis, DLS as good as it was, NTAs analysis, and we got kind of a consolidated picture that with the SUX measurement and with this microfluidic device, we are able actually to discover this early event. We can describe it, and hopefully we can actually also then mirror back and bring this knowledge back to new particle designs. So the outlook, and we have already first evidence, we are able actually to see and monitor also what happened with more complex particle system like these core shell and surface modified particle, which has single building blocks embedded in a matrix and additionally are functionalized. So we can see and follow what happens in regard what the shell is doing, what are the the surrounding proteins are doing. What we are heavily now working is to increase the complexity of the buffer system. I mean, you're probably all agree with me that albumin alone is not really a complex biological environment. So we want to go and push here the limitation to use, hopefully sooner or later, a more complex system, which is more closer to full blood situation. And my personal dream would be also that we could also transfer this type of analysis in the in situ analysis of particles in tissues in order to follow or confirm this biotransformation also in tissue. So having said that, I think my time goes to the end. So I hope I could show you a very nice label-free and non-destruction tool, which allows the monitoring of this very early biotransformation event, which I'm pretty convinced that these are important first step in the fate of particles. I think we have a proof of concept that this nanoparticle, as soon as they have the same size as the proteins, that they become a different dynamic behavior in biological environment compared to their bigger counterparts. And maybe future will show if the general protein corona concept, as it was described in the meanwhile 10 years ago, maybe has to further improved or adapted for nanoparticles which are smaller than the proteins. And with that, we hope that we can actually provide here better analytics, which allows us also a better understanding of this dynamic process on the nanoparticle biointerfaces, and hope that we can further enabling the rational design of innovative nanomedicines. With that, I would like to thank to my team, in particular to Neda, here with the glass of beer. That was the celebration of her second paper during her thesis, where we have kind of confirmed the proof of principle. In this particular case, also the SNF funding this project, and my colleague at EMPA, Antonia Niels, giving access to the X-ray center at EMPA. And last but not least, thank you for your attention. Marieluise, you are still muted.
Sorry, thank you very much, Peter, for the nice presentation. I have not received any questions in written format. Any questions from the audience, from the colleagues at present?
Maybe one quick question I would have, Matthias Wacker, investor. What would be the next step? Are you planning for serum, or do you plan for plasma, or is it really going full blood? Because actually those measurements are quite heavily doable in full blood, I would assume, right?
Indeed. So we approach stepwise. So we are planning now to increase the proteins at least with 5, 6 different proteins, and to see in how far the measurement is still reliable, and then stepwise going maybe for cell culture media, where we have controlled FCS in different percentage, and then hopefully we could learn enough to go then slowly to the plasma, and maybe one day also to full blood. But this is a journey which is probably using more than one additional PhD.
Yes, sir. Thanks.
Might I ask one more question?
Of course.
Okay. How much is the binding influenced by dynamics? Because what you are testing is a static system. When you are in the bloodstream, there will be other dynamics because you have a flow. For example, aggregation usually is not a problem. Even when you have particles which are precipitating in solution, once you have the stream, they are so quickly distributed that this is not a problem. And then there are different types of binding taking place in the bloodstream in a more complex environment.
So we are already having a certain flow. The flow is rather low, a few microliters per minute. This is due to the fact that we have not endless reservoir of proteins and particles. But of course, if we would increase the speed, we also would expect that probably the dynamics might be slightly different. But the fundamental already here is that we are not static. So we have kind of the chance to mix that in an early way, like you would inject a nanomedicine in a bloodstream. Of course, brutally simplified. But I think these are investigations we need in order to complement our classical biochemical analysis, in order to see how this biotransformation, in particular these early events of biotransformation, might trigger something we want or we don't want or we do not understand so far. So in this regard, I hope with this type of analysis and maybe a smarter design of particles than the pure gold particle, we can learn a lot in these early events.
So thank you. So then we're leaving Europe and going to Singapore. Probably nice weather over there, I suppose.
Yeah, all the time, all the temperature.
All the time, quite humid. Okay, so we are welcoming Professor Wacker in Singapore. He is working at the Department of Pharmacy, Faculty of Science, the National University of Singapore. So it's all yours.
Thank you very much. And with this, I share my screen, and we basically have to check first if there are any technical hiccups. Can you see my screen, and can you see it also?
It is not a full screen at the moment.
Yet now?
Yes.
Okay. Yeah, thank you very much for the kind introduction. So I first of all wanted to also thank the organizers for inviting me here today to talk about nanomedicine ex machina design strategies based on clinical relevance. And for those who are not familiar with this terminology, clinical relevance, we are talking about, or I talk about bioprediction today. But before we dive into this topic, I just wanted to basically highlight two trends that have been highlighted before also by the other speakers, and that is how drug delivery basically changed over the last year. So, of course, we have today more precision medicine, which means that we try to become more target-specific, population-specific, and patient-specific. But also in the drug manufacturing area, there have been a lot of changes, which affects also our drug delivery system. And that also affects how we make predictions of clinical trials. And that is the topic that I'm going to talk about today. So for those who still don't know what bioprediction is, I provide also a definition. Biopredictive performance assays provide a forecast of pharmacokinetics, and clinically relevant assays predict the first in human studies. That is the basic idea of this discipline. So how has bioprediction changed under the impression of those huge trends that I pointed out? In the past, I would say the major paradigm that we all the time had was the one of biorelevance. So we basically tried to mimic a particular administration route, for example, subcutaneous tissue or intravenous injection. And then we tried to mimic the environment as much as possible at this particular administration route. And then we kind of explained based on that the pharmacokinetics. And that was totally fine during that time, because at that time we had a lot of blockbuster drugs. We basically had also a good evidence that one size fits it all works here. But bioprediction has become more formulation-centric because formulations changed quite a bit. And that means that today what we are looking into is first the formulation characteristics. For example, you have a liposome, then you probably have to consider the presence of lipases at your administration site. And then you look into the observed in vivo responses. And in drug development, we of course have to run in vivo studies. And then you make mechanistic considerations. How could this particular effect be on the pharmacokinetics? What could have caused this effect in the pharmacokinetics? That leads us to the so-called biologically relevant attributes and the physiological triggers, for example, protein concentration that we heard about, and maybe also the mechanical properties of the proteins. So, but how does that relate to a design strategy? And that here is a nanomedicine production process that you will probably find today in Singapore, in our lab, or also in other labs. You have a nanomedicine production, you use a bioreactor system, and then you produce, for example, extracellular vesicles. You can do that by changing temperature and medium saturation. Sometimes extrusion is involved. And then you load into those carriers drug substances. And what you get is a drug-loaded vesicle. But that is actually not really a design strategy. That is only a nanoparticle design. It only becomes a design strategy when you are really able to make educated decisions. What kind of drug release, what kind of encapsulation, what kind of particle count, and what kind of vesicle composition do I need in order to get the pharmacokinetics that I need later in my patients? And that is very much what bioprediction is about. We try to estimate the clinical performance using those measurements. And how do we do that? We very much focus on drug release, because so far we can say, because in the clinical setting, we measure usually the plasma concentration of the drug substance, the drug release is still our strongest indicator. This is what we measure clinically in the phase 1 clinical trials. So we also have to measure it in vitro. And when I started with this research around 2010, there was not actually a lot of methods to measure the drug release. There was one commercial product, and that was, sorry to say, poorly standardized. So what we did is we built our own equipment, and now you can also see that this dispersion releaser cell, this here is a USP apparatus with a release cell based on dialysis, from which basically this pink liposome releases its compound. We built this cell and built it into a USP, United States Pharmacopoeia dissolution tester, in order to also follow international standards. Because from the perspective of a drug company, of course, it makes no sense if you have a method which only works in one lab and not in others. So now that we are able to also measure the drug release very much, and I can really say we set standards here, we also basically included all the validation protocols for this equipment into a stimuli article for the USP, which means we hope also that soon our equipment will appear as an international or at least national standard first in the United States. So, but how can we use this measurement now if we are designing nanomedicines? And I think our nanomedicines are not produced differently from the ones that you produce in your lab. We have microfluidic technologies here. This here, for example, is a herringbone mixer, and we also use that. Those are 3 different types of polymer nanoparticles. I think this is a polymethacrylate, PLGA, and PLGA PEG. But we also integrated a lot of measurements which were available at the National University of Singapore already. So a dear colleague of mine, I hope she's in the audience, Georgia, she developed those hybrid vesicles, which basically are a combination of synthetic liposomes and a cell extract, and also those biotechnological methods. We kind of adapted and use them now in our bioprediction approach. So how do you start? First of all, I started by finding someone who has clinical data, and that is usually with the companies. And this company here brought 2 drug products to the market. One is Phoscan, and the other one is Phoslip, both of a poorly soluble photosensitizer. And the first thing that you have to go when you want to explain pharmacokinetics is really understand what's happening. What is your data leading you to? And in this study from 2019, we were able to really very much predict the pharmacokinetics based on 2 parameters that you can measure and easily assess. That was the drug release, and that was the particle size distribution that we measured here by NTA. And you can see that the prediction is already very nice. We also did, for example, the particle stability here in serum. So because we also wanted to measure that, that is also where the question came from. And yeah, in this case, we were able to make basically an estimation what would affect the pharmacokinetics mostly here. The next step was that we had a liposomal formulation, and of course the company also had the in vivo data there. And we didn't really use any in vivo data for this liposomal formulation. We made a prediction based on what was preclinically available, which means we looked into the release, the particle size distribution and stability. And we looked also into basically the early mouse studies that are usually available when you translate a liposomal formulation into the next stage. And our prediction was, again, very much accurate. So you can see here this line, this bluish line somehow, which basically very much overlaps also with the pharmacokinetics. Of course, that is not really population specific yet, but it is at least a good hint what kind of information is you have to put into your formulation to design the pharmacokinetics in a particular way. So what did we do next? I also want to explain a bit behind the scenes how we did that. So you start with a pharmacokinetic model, and in our case, we also try a very robust approach, but that also means that we are not detailed. Because when you think about it, in the clinical trials, you do not measure to which cells exactly your nanoparticle system goes. You just measure drug concentration. And sometimes you also measure how much was maybe in the blood cake and how much was basically in the plasma. So you have to rely on what you really know about that, also on the kinetic side. And that is, you have basically an infusion rate that, of course, you know. Afterwards, you have the carrier bound fraction. And this is safe to say also from the preclinical data that we have that most liposomes stay there. But if they leave also the vasculature, then they do not return. They do not, it's not really like a distribution henceforth. It's basically if they extravasate, then they also stay there. It's basically just a statistics game there. And then we, of course, have also the drug release, because the moment you put your drug formulation into blood circulation, you have an enormous dilution, and that automatically drives your drug substance out of your carrier. But we didn't really build that up only based on phospholip, because we had a model that we thought could be also be generalized for other nanomedicines. So we did a validation using ambisome, doxil, phospholip, the one that you already heard about, and Myoset, which are all commercial formulations. And we also included a polymer nanoparticle formulation, which was an investigational clinical candidate to explain with this kind of model the pharmacokinetics well. And in all cases, it worked very well. So if we now try to also put that together, what can you do in order to establish such a correlation and later use it also for bioprediction? And here I want to caution you, always listen to the explanation when we come to the extrapolation. I just say that early. So the first thing that we did is we used two formulations that are really hard to establish IVIVCs for in vitro, in vivo correlations. That was Lipodox and Nanocore 7.4. One is a liposomal formulation, a pegylated liposome, just like Doxil. It is the generic version from Sun Pharma in India. And then we had from a collaborator, Nanocore 7.4, which is an investigational drug product, so a clinical candidate, which was tested in a small clinical trial in Russia. So the first thing that you have to do, you establish the correlations. And those are the two correlations that we got when you use a dedicated pharmacokinetic model. Together with this information about the drug release, you can design now such an in vitro, in vivo correlation. The next step that you have to go then is you have to basically generalize that, and you have to apply that to new formulations. And this is where it becomes tricky. Our two formulations were Nanocore 6.4, which is another candidate from the same line like Nanocore 7.4. And the other one was an extracellular vesicle preparation, so dramatically different also from Lipodox. Still, we created now estimated pharmacokinetics because, of course, the model can do that. You just enter the in vitro data, and then you now get also an estimation of the in vivo data. But I said, please listen to the explanation because that's important in this discipline. Nanocore 7.4 and 6.4 are almost identical formulations. The chemistry is totally identical. The only thing that differs a little is the salt form inside the nanoparticle core and the drug load, which means you can expect that Nanocore 6.4, this formulation here, this estimation, really very much reflects what you will also later find in probably a clinical trial, because the only thing that differs is the drug release in this case. So we can probably now suggest to this company a way of improving their formulation by triggering in a particular way the drug release. Now we look at the second estimation. In this case, we have a totally different particle system. The vesicle is different, and that is what mostly comes in contact also with the environment, with the physiological environment. So we cannot really say if, for example, the particle clearance of those vesicles will be much, much quicker because the carrier will be eliminated, for example, by cellular uptake. But what we still can say is that even if you find now an extracellular vesicle which circulates for 1,000 hours, probably you will not measure drug concentration for more than 50 hours in the bloodstream because the release behavior is still valid, in a valid way, simulated in our assay. And that basically means that because the drug release will be too quick, you probably have then an empty carrier which circulates for 1,000 hours. And that would be a problem in development. So here we could suggest, do not only optimize the circulation time, also look into the drug loading procedure, because this is what also drives your pharmacokinetics significantly. So to sum that up in a nutshell, bioprediction becomes more formulation-centric. I hope I was able to convince you of that. We have more diversity in the drug delivery market. When we do those estimations, then we require a good understanding of the processes that occur after administration in humans. I underlined in humans because very often we also draw conclusions from an early mouse model. We found that the pharmacokinetic data that we need is at least red, otherwise we cannot really work with it well. And bioprediction streamlines nanomedicine optimization in the last steps from bench to bedside. That is also why we very often do that together in collaboration with a company, because release technology is one of the typical benchmarks that you have to do when you also apply for approval with a particular drug product. So in the end, I want to thank all my collaborators. And here in this short video that we made during the COVID break, as you can see from the masks, this is the team that was involved also in collecting this data. They made a very nice video, and since then I show it at every conference. Thank you very much, and if you have questions, I'm happy to answer them.
Yeah, thank you very much for your presentation. We have questions from the audience or from the colleagues. Okay, good. So then we go from Singapore back to Groningen, in that case, to Professor Anna Salvati. So her lecture today is identification of corona proteins, mediating nanoparticle uptake and characterization of the mechanism of endocytosis.
Good morning, everyone. Can you hear me properly?
Yes,
Perfect. Thank you, and thank you also for the opportunity to present some of our work today. And I would like to start with this to thank the people involved. You have heard Roberta from the earlier talk, some of the work we do. I will continue from what Roberta introduced to you, and a lot of what I will show today is actually also work of Valentina, who's a former group member and now is here there in Basel University, and you will see the name of the other people involved and the manuscripts that refer to the work I'm presenting. So our group is interested in understanding how nanoparticles, nanomaterials are processed by cells, with the idea that with a better control of that, with a better understanding of this mechanism, we can design nanomedicines with targeted behavior. And in particular, we're interested in the early interactions at the cell membrane and the subsequent mechanism of internalizations, with the idea really that ideally we would like to be able to change material properties to control the outcomes at the cell level. So what have we learned so far about the initial interactions at the cell membrane? And of course, in nanomedicine, we try to use active targeting, molecular targeting. We try to attach ligands on the surface to specifically recognize the targeted cells. And we know that that's extremely challenging for many different reasons. It's not as simple as attaching an antibody. And we also know that once the nanomedicines are applied in biological environment, we have already heard that today, there is a corona formation on the nanomedicine and nanoparticles, and this layer changes completely the interactions with cells. There are different strategies to try to avoid it or reduce it, but more and more we're discovering in the field that in some cases this adsorbed layer can actually provide new interaction to the nanomedicines. And the most surprising example perhaps is that of approved nanomedicine on Patro, the first sRNA nanomedicine, which can target the liver hepatocytes in vivo because it recruits ApoE in the corona. So then the interest in this layer changes, and now we can see new opportunities in understanding better how this layer modulates the interactions with cells and even in vivo distribution. And Roberta already mentioned to you, we use often this example of another nanoparticle interacting with the LDL receptor. We have the silica nanoparticles that after corona formation, similar to the Onpattro example, because of the corona can interact with LDL receptor. But the story is actually more complex because this depends on the details of the corona. And we know that when we incubate particles, when we expose particles to different serum concentrations, different coronas can form. You see this at low serum concentration, like in vitro condition, 10% serum, though it's human in this case. And if you expose the same particles at higher serum concentrations, different corona forms. And if you silence the LDL receptor, we see that only the particles with the corona form at high serum concentration is the one capable of interacting with this LDL receptor. So the same nanoparticles, depending on corona condition, can engage with different receptors in the same cells. This is important to keep in mind. What about uptake? What about the mechanism of uptake? This is a topic I love. I love endocytosis, and there are many different methods to study uptake, and it's not easy. No method is perfect. Every method has different limits and problems or benefits, and we've done a lot of careful optimizations to try to use these methods without creating artifacts. Roberta mentioned the work she did together with Daphne with full genome screening. We can use proteomics and organelle flow cytometry, live cell imaging and colocalizations. But of course, there are also classic methods to study mechanism of uptake using transport inhibitors and silencing to reduce the expressions of key proteins involved in endocytosis. And yeah, with appropriate optimization, we try to use combinations of these methods to understand the mechanism of uptake. So going back to these particles at low and high serum concentration, which interact with LDL receptor, we know that LDL receptor typically is internalized by cells via clathrin-mediated endocytosis. And as Roberta anticipated to you, actually we can block clathrin-mediated endocytosis, but the particles interacting with LDL receptors are not affected. At low serum concentrations, we see an effect, but high serum concentrations where there is the LDL receptor involvement, the uptake seems not clathrin-mediated. So this tells us two important things. First, that the same nanoparticle, that the corona affect also the mechanism of uptake. So depending on the corona, we have different receptors interactions, and the same nanoparticles enter the same cells with different mechanisms. So we really have to be asking which is the relevant corona to use then for our experiment, since it affects receptor interactions and also mechanism of uptake. Second, even when we target a receptor, let's say that here we are targeting the LDL receptors via the corona in this example, even if we engage with specific receptors, it doesn't mean necessarily that the cells process the nanomaterial in the same way as we know for that receptor. So in this case, at high serum, even if LDL receptor is involved, we have a sign that uptake is not clathrin-mediated. So targeting is not easy, but it's also does not imply if I target a receptor, it doesn't mean necessarily that then the cells internalize the nanomaterial in the same way as that we know for that receptor. But we really wanted to be sure of this LDL receptor condition. We really wanted to be sure that uptake is not clathrin-mediated. And there are different methods to try to exclude that, to try to confirm that. This is another example. You can overexpress these proteins, AP180, and the cells becomes green. Transferrin is a protein, another protein that enters via clathrin-mediated endocytosis, and you see the green cells do not uptake transferrin, while the non-transfected cells have a huge transferrin uptake. And you do the same with these silica nanoparticles at high serum concentration, and in the green cells and in the black cells, so with or without clathrin-mediated endocytosis, uptake is the same. So this is another confirmation that the uptake seems to be not clathrin-mediated. And you can also silence clathrin and other proteins involved in this pathway. And transferrin uptake in this condition is, again, very well inhibited. We have all the appropriate controls, but again, we don't see evidence of clathrin-mediated uptake for the nanoparticle. And then we start to see that many different proteins involved in many different pathways seems to have a role. Partial role in the uptake. So can it be so complex? We have seen from Roberta different receptors. Here it looks like there are multiple mechanisms. So is it really such complex mix of interactions, a particle engaging with multiple receptors and internalized by cells by multiple mechanisms? So are we thinking it in the wrong way? Can we strip it to the basics and make it as simple as possible? And there are some key aspects in endocytosis, if you want to strip it and simplify it as much as possible. There are common features of endocytic pathways. There is a first interaction at the cell membrane with specific proteins, and also the lipid of the cell membranes are very important. Then typically there is some mechanism for bending the cell membrane to allow invagination of the membrane and the formation of vesicles in which the material is internalized. So let's forget what we know about all these other mechanisms of uptake. What can we say about mechanism of membrane curvature generation in nanoparticle uptake. Nanoparticles are big, and they are known to be capable of bending the cell membrane, and we know that the cells has different mechanisms to sense membrane bending and assist membrane curvature during uptake. So we were interested to study the role of these curvature sensing proteins in the uptake of nanoparticles, knowing that particles are big, can bend the cell membrane. And some viruses and pathogens induce their own uptake by bending the cell membrane and activating and recruiting this kind of curvature sensing proteins. So we selected a series of bar domain proteins. Many of these proteins contain the specific bar domain that is capable of recognize the curvature of the membranes when it happens. And there are many of these proteins, and several of them have been described for a role in different mechanism of endocytosis. And here we just wanted to screen a bit. We selected a panel of them and try to see, do they matter for nanoparticle uptake? And as you can see, again, for many of them, we see strong reductions of uptake when we silence their expressions. And if you change particle curvature, in this case by changing nanoparticle size, so the larger particles locally have a lower curvature, you see that the role of these proteins change with curvature as well. So this is just the beginning. I think there is much more work to do, of course. It's just the first indications, but what we're trying to do is really trying to dissect this mechanism and keep it as simple as possible. First, receptor interactions, and then perhaps because the particles can bend the cell membrane, we want to understand more of the mechanism of membrane curvature generations and the involvement of these specific proteins in the mechanism of uptake. So more work in these directions in the years to come, but we hope to manage to disentangle this mechanism. Now, back to the corona and the cell receptors interactions. Having seen that these corona layers can mediate specific interactions with cell proteins, of course, there is interest in trying to use it. And one thing that is happening in the field, and we also tried a bit, is we change nanoparticle design. These are liposomes of different charge. We know that we form different corona, and then we can screen for nanoparticle uptake and do a correlations between uptake efficiency and corona composition to try to identify corona proteins that mediate uptake. So in this example, we use liposomes from neutral to more and more negative charge. We use size exclusion chromatography to isolate the corona-coated liposomes, and mass spectrometry to identify the composition. And as expected, you change the charge in this case, and different coronas are formed. And of course, also different uptake kinetics are observed. So now we can correlate uptake and corona composition to try to identify proteins that promote, or also proteins that reduce, corona proteins that promote or reduce nanoparticle uptake. And this is happening more and more in the field. This is an example with liposomes. We did it also with another panel of particles. In this case was silica of different sizes and different functionalities. Different coronas, of course, when you change size and when you change charge. And in this case, we use endothelial cells because they're relevant for nanomedicine. Endothelial cells derived from different organs, and compared uptake kinetics of these 6 nanoparticles in 4 different endothelial cell lines. So we have a panel of uptake and corona, and then you can do correlation analysis. And this is the kind of data that you find. Of course, there are different coronas, and you can try to identify, for instance, in this example, we found that these proteins. In literature, there are other examples of this. This protein is very abundant in the corona, and we saw that it's negatively correlated with uptake. So the higher the abundance of this protein in the corona, the lower the uptake. But now correlation doesn't tell anything in itself. We need to validate that it's actually something that mediates an effect. How to do that? We can create an artificial corona. We can start to engineer the corona. And for instance, we form the corona only with this protein, and compare uptake of the bare particles or the full human plasma corona with the uptake of the artificial corona based on these correlated proteins. And what we found in this example, we did that with different proteins for which uptake was correlated negatively or positively. You need to do this because in some cases you see that correlation alone doesn't mean anything. But in the example of the HRG, we could really see that engineering the corona, creating an artificial corona of HRG, the uptake was reduced in the same way as when we were doing the full human plasma. So this protein alone seems to be capable of reducing uptake of nanoparticle in this case. So this is it for today. There is a lot of ongoing work to keep understanding how the particles are internalized by the cells. We're interested in really understanding the molecular mechanism of endocytosis so that we can ideally control it. And definitely we need to take care also of the early interaction of the cell membrane, which corona conditions we use, because these can affect receptors interactions and mechanism of uptake as well. And hopefully with better understanding of all of this, we will be able one day to control it and fully exploit it in its potential. And thanks again. And these are the people involved and the name of the people that helped me with this. And thank you all for your attention.
Okay, thank you very much, Anna Salvati. We have questions to her. Still too early for questions? Okay.
No, I would have.
Okay,
I would have
good.
one.
Good.
Anna, as usual, very nice presentation, excellent work. May I speculate with you together? What would happen if you downsize your particle system to 5 nanometer or lower? Would you still have these apolipoproteins attached? And what would be with the biokinetics?
As you said in your talk, it will be very interesting to see when the protein size becomes comparable to the nanoparticle, what happens. We have the first examples. There are works with smaller particles, and we have to see if these classes of proteins are still relevant or not. So the short answer is yes, it's very interesting to study it, but I don't know. And, actually another question is also if I'm looking at the proteins, but many works have shown that it's not the protein only that binds, but the full lipoprotein particles, so both the lipid and the proteins assembled. So you have a small nanoparticle attaching to the nanoparticle. So we also have to identify exactly how the material rearrange once the nanomedicines interacts with these lipoproteins. So we talk about apolipoproteins, but is it the apolipoprotein at all, or is the full LDL, HDL nanoparticle that is sticking and interacting? And this becomes even more relevant when the sizes are comparable. I don't know, it would be interesting to see.
Thank you.
In terms of curvature, it might matter even more for this curvature and induction mechanism, but we will have to test it, yes.
Thank you.
Okay.
Oh, sorry. Can I ask one more question?
Yeah, of
yes,
course.
please.
Okay. Did you check with spheroidal cell system instead of a 2D cell system?
That's another thing that we're doing now. Absolutely. We know that the biology of the cells can change a lot. So definitely going in 3D is the next step, as also going in a dynamic system with presence of flow that is essential both for, especially if we look at endothelial cells, both for the cells and for the corona interactions. So those are all examples, all directions that we're exploiting now in the lab, because I expect things to be different in a 3D structures. Actually, Roberta did quite some work with tissue slices, so we can also use tissue slices to study this mechanism, but we also need to develop methods to study endocytosis in this kind of 3D systems, and that's another challenge. So when you're interested in studying the mechanism of uptake in a 3D systems, we first need to do quite a lot of optimization. And she used tissue slices, so a full 3D slice of tissue. And really, we need to implement new methods also to study these questions in more complex systems. Similarly, in vivo is still quite difficult to do it directly in vivo. But we need to arrive there. So definitely that's an interesting and important direction.
Okay, so then we have a last short presentation from Q Quinn from London.
Yeah. Hello, everyone.
Hello.
Hello. Can you hear me?
Yes, we can hear you, I think.
Okay, so I'm going to show my screen. Can you see my screen now?
Yes, but it's still small. So if you can enlarge it a little bit.
Oh, yeah. I think that's fine now.
Yep.
Yeah.
Thank you.
Okay, thank you for your introduction. Good morning, everyone. My name is Yue Qing, a final year PhD student from King's College London. It's a great pleasure to have my first talk in Castellano during my PhD. And my topic is optimization of stable nucleic acid lipid nanoparticle formulations for in vitro and in vivo delivery of plasmid DNA and siRNA to cancer using design of experiment. And we all know that the unexpected immuno-related adverse event limit the application of monoclonal antibody-based immunotherapy. And we found the therapeutic nucleic acid is an alternative to antibody for cancer immunotherapy. Therefore, in this study, we would like to use stable nucleic acid lipid particle SNAPs to co-deliver the plasmid DNA and the siRNA for cancer immunotherapy. SNAP consists of 4 lipid components, and the lipid composition could affect the nucleic acid delivery efficiency. And then we found the design of experiment. DOE is a potent approach for formulation optimization. Therefore, our aim in this study is to optimize lipid composition for SNAP formulation and then to obtain the optimal SNAP formulation that can co-deliver the plasmid DNA and the siRNA in vitro and in vivo for cancer immunotherapy using design of experiment. And the novelty of this study is we know the DOE was mainly applied for the mRNA and the siRNA delivery, but not for the plasmid DNA. And also DOE was mostly applied for the optimization of formulation making processes, but not to study the impact of lipid composition on nucleic acid delivery. And in this study, our SNAP consisting of these 4 lipid components and was formulated with either siRNA and the plasmid DNA by ethanol dilution method. Then we put these 4 factors and the 3 responses to the DOE software and to establish 24 different lipid composition SNAP formulation. For the transfection evaluation, we use the luciferase reporter gene PLUX and the SLUX SNAPs to transfect the non-luciferase expression model and the luciferase expression model respectively. Then all the data were put into the DOE software to establish the predict model and then to obtain the optimal lipid composition. And here are the results of the size, PDI, zeta potential, encapsulation efficiency, in vitro and the in vivo transfection efficiency of PDI and the siRNA SNAPs. Then we put all this data into the DOE software and we get this contrast. Today, I'm just going to show you the PDI SNAP as an example. The contrast here present the effect of KC2 DOPE cholesterol lipid compensation when fixed the PEG at 1%, 3%, and 5% on size response. We can obtain the big size at the red area and the small size at the blue area. We can see the blue area expanded when we increase the 5%, which indicate the size will decrease when we increase the PEG percentage. And similarly, for the encapsulation efficiency response, we can see when we increase the PEG percentage, we also can have higher encapsulation efficiency, which is the red area here. And for the in vitro transfection, we only can obtain the highest pDNA transfection at 1% and the 5% PEG, which is the red area shows here. And for the in vivo transfection is the same. We also can get the highest pDNA transfection at 1% and 5% PEG. And the heat map here showing the correlation between the lipid composition on different size on different responses, and the blue color indicate the negative correlation, and the red color indicate the positive correlation. From this heat map, we can see the lipid composition could affect the size, in vitro and the in vivo transfection efficiency, both in plasmid DNA and the siRNA SNAP formulations. And the conclusion we can get from this study is the design of experiment approach allow us to select a middle ground SNAP formulation, which can achieve high PDN transfection and gene silencing in vitro and in vivo. And this will in our future study to simultaneously deliver therapeutic siRNA and PDN for nucleic acid-based cancer immunotherapy. And finally, I would like to thank my group members who helped me in this study, and also thank you for your listening.